All right, welcome everybody. I realized that in my last introductions I didn't introduce myself. So this is Manuel Rausch. I'm an assistant professor at University of Texas at Austin, and I'm a co-host of the Closer Look Open Journal Club in Biomechanics. Our speaker today is Dr. Kyoko Yoshida, and I'm not sure how her job search is going, but potentially future Professor Kyoko Yoshida. And she'll be talking today about a project that she's been working on during her postdoc on multi-scale models of pregnancy-induced heart growth, integrating hormonal signaling and mechanics. And I also want to take a quick moment to let everybody know we have another couple of talks coming up soon by Matt Bursey, Jacopo Ferrucci, Nele Famey, and Sara Roccabianca. So, stay tuned for those as well. So about Kyoko real quick, Kyoko got her PhD at Columbia University with Kristen Myers, who most people know through her work on the biomechanics of the reproductive system, which obviously is really important work. And what I really appreciate about Kyoko's work now is that she really brought sort of her past training and interests into the work during her postdoc with another person that most people know, Jeff Holmes at University of Virginia, who obviously does a lot of cardiac modeling. So I think your title makes it pretty clear. You bring together these two interests, and I think that's super fascinating. Kyoko and I actually met, I think the first time at a workshop for FE Bio, all the way back, maybe 2015.
Yeah, I think somewhere around there, yeah.
Park City, Utah. And I think you had just started mountain biking, which as I can tell from your online representation is now an intense hobby of yours. So yes. That's pretty cool to see as well. Anyways, with all this being said, Kyoko, I'm extremely happy for you to be here today and to continue our tradition of excellent speakers in this Biomechanics Journal Club. And I'll hand you the virtual microphone with that.
Sure. Well, thanks so much, Manuel. That's very kind of you to say. So in terms of job search, I have accepted a position with the University of Minnesota. So I'll be starting a lab there this summer. So if you're an undergrad and you're interested in pregnancy biomechanics, email me. And then another note about kind of like studying reproductive biomechanics in my PhD and heart mechanics in my postdoc. So this is really the project that like I had been envisioning, at the beginning of my postdoc. So it's actually like, I'm really excited about this paper and I think you'll be able to tell. So I'm really looking forward to a great discussion. Please feel free to interrupt me during the talk if something is not clear. I'm very comfortable and happy to do that. And I don't have too many slides because I really wanted like people's questions to kind of guide the discussion. So with that, I'll go ahead and get started. And so to start with, what I think to start with is that like pregnancy really stands at this interface between mechanics and biology, where we're seeing huge changes in both. So on the mechanics side, we have increased loading and growth of virtually all maternal soft tissues as the baby is growing. So today I'll be focusing on the heart and the circulation system, so the mother circulation system. So in her circulation system, her systemic resistance actually drops by 34% over 9 months of pregnancy, and her total blood volume actually expands by 50%. And in response to this, the heart actually does a lot more things. So it actually pumps 45% more blood during pregnancy. So her cardiac output will actually go up. And it also grows about 20% in size. So her heart actually does grow throughout the course of a pregnancy. And if you're really interested in this topic, I want to highlight a couple of papers here, especially this review paper at the bottom that does a really nice, it's a chapter that does a really nice job of kind of describing all of these different changes that are occurring in the maternal cardiovascular system. And then, of course, in the biology side, we have these surges in the pregnancy hormones. So we hear about pregnancy hormones all the time. So in particular, I'll be focusing on two of the main hormones, estrogen and progesterone. And here I'm just showing how their levels increase throughout the course of a human pregnancy, so over 42 weeks of pregnancy. So you can see about a 10 times of an increase in both. And so pregnancy is actually described as a cardiovascular challenge for women. And so here's that cardiac output that I was talking about before. So in the early stages of pregnancy, your cardiac output is going up. So it's almost like the woman is actually exercising even when she's not. And then it kind of stays level. And then after birth, it actually drops back to the non-pregnant state after 2 weeks. And this to me was really interesting plot for me as a biomechanics person, because this is like increased loading, kind of level, and then this sudden drop at the end of the pregnancy. And this drop is actually, I won't be focusing on that too much today, but it's actually really important because a lot of the problems that can happen with the heart during pregnancy actually happens in this postpartum period. And so there was also a recent paper in Science Advances in 2019 that was published that actually compared pregnant women to like ultra elite endurance athletes, and the metabolic demands are actually like not that different. And so, yeah, it's a huge challenge for women. And then I just wanted to show you how the heart grows in size. So these are images from mice, so a non-pregnant mice, the whole heart here, and then in late pregnancy. And then if you were to kind of cut across and look down at the cross section, you can see the LV here, and then the right ventricle here. So I'll be mostly focusing on the left ventricle today. And then you can see at the late pregnant stage, it's grown in size. So why is this important to study? So I kind of alluded to this in the previous slide, but heart problems are actually the #1 causes of pregnancy-related deaths. And then that was coupled with the fact that maternal mortality is actually rising in the United States, especially compared to other developed countries. And I also wanted to point out that this week happens to be Black Maternal Health Week. So if you're interested in this really important topic, I would really recommend you to look up the hashtags Black Maternal Health Week and BMW21 on Twitter. There's actually been really interesting resources and important conversations about that lately this week. And so we can look at reasons why mothers are dying. So if we plot the causes of pregnancy-related deaths kind of from 1987 up until now, you can see that we're doing well with treating certain conditions. So hemorrhage, so this is when a mother undergoes uncontrollable bleeding after delivery, hypertension, or high blood pressure and infection. So these rates are going down. So we're doing pretty well at treating those conditions. But then the rates of cardiovascular conditions and cardiomyopathy, so this in particular is heart failure associated with pregnancy, is actually rising each year. And they make up for, together they make up almost 1/4 of the reasons why mothers are actually dying related to pregnancy. And so the research question that I'm really interested in is, because we have all of these big changes in the mechanics and also in the biology, how are these signals interacting to drive this really important growth and remodeling process? So I think we can all appreciate that mechanics does a lot to affect the tissue growth and remodeling process. So we know that if we stretch on a cell, or like I guess the most obvious example is like if you go to the gym and you lift more weights, right? So if you increase a load, then your muscles are going to react by growing. So we know that mechanics can influence this process, but we also know that hormonal signaling also has a role in this process. And so this tissue growth and remodeling process then can actually feed back to change the mechanical states within the tissue. So for example, if your tissues grow in size, then the cells are probably feeling less stretch. Or if your tissues are getting softer or stiffer, then the stretch is also changing as well. And so this feedback loop is also really important to consider. And so this is really the schematic that I'm really interested in developing, not just in the heart, but also in other reproductive tissues as well. And so I wanted to talk a little bit of background about how hormones and mechanics actually influence heart growth. So I mentioned 2 hormones, estrogen and progesterone. And so estrogen has actually been shown to prevent or limit heart growth. So here I'm showing an experiment where in mice, so this is a controlled mouse heart, and you can infuse mice with high levels of hormones. So this is actually angiotensin 2, which is not necessarily a pregnancy-associated hormone, but more of a heart failure-associated hormone. And when you basically like infuse mice with this hormone, what happens is their heart actually grows in size. So you can see that the size of this heart is bigger than the control heart. But then when in this experiment, they infused angiotensin along with estrogen and found that the heart growth is actually attenuated. So you can see that the size of this heart is smaller than the angiotensin 2 heart, more comparable to the control heart. So estrogen seems to have a role in limiting heart growth. On the other hand, the other hormone, progesterone, has been shown to induce growth. So this has been shown with one group. So progesterone is really, really understudied. So if you're interested in the heart, I would really recommend you, and please talk to me about the roles of progesterone on heart growth. But in these experiments, so they did an in vitro experiment where they took out cardiomyocytes, so heart muscle cells, and then they treated them with progesterone. And you can see in the right image that each of the cells actually got bigger. So they quantified about a 50% increase in the cell area. And the same group also repeated the study in vivo in mice and saw that heart mass grew by about 10%. So if you infuse mice with progesterone, their heart also responds by growing. And then in the cardiovascular biomechanics field, we can all appreciate that mechanical overload also induces growth in the heart. And so pregnancy is usually associated with a volume overloaded state, so that increase in cardiac output. And so here I'm showing an experiment in rats where they induced volume overload and they observed about a 2-fold increase in heart mass. But what was interesting to me in this study is that they also looked at the combination of volume overload and estrogen, and they saw that this heart growth was actually attenuated. And so you can see that the blue bar is lower than the red bar. So you can see that this group actually did not grow as much. And to me, this is really interesting because this is showing us experimental evidence that mechanics and hormones can actually interact to influence heart growth or to change heart growth during pregnancy, or not during pregnancy, but just in general. And so with all of this in mind, what I've developed in this paper is a multi-scale heart growth model for pregnancy. So the idea here is that we're looking at these interactions between the hormonal signaling and the mechanical loading. And so I'll be talking a little bit about the cell signaling network model. So this is what's shown on the left hand here. And so the big idea here is that we can use these types of models to take into account the changes in hormones that are occurring during pregnancy. So that rise in estrogen, as well as the rise in progesterone. And then we couple this to a mechanical growth model. And so the big idea here is that we can use this model to take into account the changes in mechanical loading that occur during pregnancy. And I call this a multi-scale model because this cell signaling network model is actually simulating all of the intracellular or signaling pathways, so all the signaling mechanisms that are happening within the cell, which we then try to translate into the cell level growth, which we then feed into this tissue organ. And I put system here because we really are looking at all these resistors and capacitors are actually representing like the aortas and the vein within our body. And so this is what I'll be outlining in this talk today. And so first I'll start with the cell signaling network model, so the left side of the image here. And so the cell signaling network model was actually built with a software called Netflix in collaboration with Jeff Sosserman, who's at the University of Virginia here. So this model is not just for pregnancy, it's actually a really cool software. So if you're interested in building models like this, I would really encourage you to check out the GitHub website that I've listed here. But again, the big idea here is that we're simulating these intercellular signaling pathways that are leading to cell level growth. That's why I have this little cell here. And so the inputs into this model are the hormones. So we have progesterone and estrogen, the pregnancy hormones. And I also included angiotensin 2, which actually rises a little bit during pregnancy, but then also in cases of volume overload and heart failure, angiotensin 2 levels go up. And then we have this mechanical signal stretch. And then so the main output that we're interested in this model is cell area. So this is really representing how much the cells are growing in size. So this represents our growth. And then the lines that you see here are all the different reactions where all of these different species can interact with each other. And so basically what this model ends up being is a system of ordinary differential equations. So these are actually hill type differential equations. So we get like a nice sigmoidal curve, which is, you know, everything in biology I feel like is a sigmoidal curve. So this is also going to be a sigmoidal curve. And these are normalized, meaning that each of these little blocks here can take a value between 0 and 1, where 0 represents that there's no activity of that particular species, and 1 actually represents maximum activation of that species. species. And so you might be wondering, how did I come up with all of these different lines? And so we actually do this by looking at experiments. So in particular, I looked up in vitro experiments. So these are cell culture experiments where cells were treated with progesterone. So that's actually the image that I showed previously, as well as treating with estrogen and the combination of progesterone and estrogen. And then angiotensin, and then angiotensin plus estrogen. And so we can actually simulate these conditions and look at different nodes that people report, like in the experiment, and see if it goes up or goes down. And we can actually adjust all of the reactions to actually match that in vitro data. So that's actually how I built them. And then these are the papers that I pulled the data from, which is also included as a supplemental document in the paper. And so that was the cell signaling network model. And so now I'm switching over to the right-hand image. So this is the mechanical model. So here we're using a compartmental model of circulation and heart mechanics. So you might usually see a finite element model, but in this particular study, we used a compartmental model, which we actually like to use in our lab. So if you're interested in that framework, I'd encourage you to look at the Wittenberg and Holmes paper from 2018. But basically what we do is we say, okay, the heart is a thin-walled sphere with a particular volume and a particular pressure, just to make it more computationally inexpensive, because I'm going to be simulating growth. And so that was kind of important to us. And I also mentioned that we've been particularly successful in using this framework to actually simulate heart growth in the context of volume overload and pressure overload as well. So we feel pretty confident in using this type of approach. But the big idea here is that we can use this model. We can simulate how the blood flows throughout the body and into the left ventricle and the right ventricle. And then we can simulate these pressure volume relationships in the heart. So here I'm showing the pressure volume loop of a control heart. So the dots here actually represent data, experimental data from rats in the Holmes paper in 2004. And here I'm just showing that we can actually adjust our circulation model parameters to actually match the experimental data. So the solid line here is showing how the model can match that data. And so the nice thing about this type of modeling too is that it's actually pretty easy to modify to represent the experiment that you want to simulate. And so because we wanted to look at the effect of increased stretch, we also simulated the case of volume overload. And so a lot of the experiments in rats for volume overload is introduced with the fistula, or a connection between the aorta and the vena cava. So you can actually add another resistor here between the aorta and the vena cava to actually divert blood flow like they would in an experiment right back into the RV, which then causes the volume overload. And then so in the red is what I'm showing is the volume overloaded state. So you can see that we can actually match that the experimental data just by adjusting the resistor here. And then I think I just also adjusted the amount of stress blood volume in the heart, in the circulation. And so this model is really telling us that, okay, so if we increase stretch by this much, then we're going to see what happens to the growth. And so next I wanted to talk about how we actually do growth. So how do we actually account for growth? And so we use a pretty typical framework, so the kinematic growth framework that's been used in a lot of different papers. And so the reason why we need this is because when we observe the tissue in vivo, before it's grown and after it's grown, the shape change that we observe is due to both changes in loading as well as changes in growth. So the heart can actually be under two different pressures. So it can be under pressure one before it's grown and in a different pressure after it's grown. But then there's also the shape change, right? So individual cells are actually getting bigger. And so this framework really allows us to decouple these two effects. And so the way we do this is we first unload the ventricle so that it's in a stress-free state. And then we apply growth. And so a lot of the papers out there have a mechanics-based growth law where the FG, or basically the growth that's occurring, is based on some metric of mechanics. But here in this model, we wanted this growth to be dependent both on the hormones and the mechanics. So it's actually a function of the cell area output from the network model. And so if we apply this, so this actually doesn't apply to the compartmental model, but if we apply this, for example, in the finite element framework, you can get incompatible growth because we're applying it at a stress-free state. But then we can apply an elastic stretch to enforce compatibility. And so here I'm just showing some of the residual stresses that can actually arise from enforcing this elastic stretch. And then we can reload it back to that grown pressure to get back to the state. And so this really lets us kind of decouple these two effects from both the loading and then the growth. And so so now we've kind of got all of the different pieces together. And so next I want to talk about how these models actually talk to each other. So I talked a little bit about the growth. And so for this paper, we just decided to do 2 times the cell area as a function of FG, the determinant of FG, or the mass increase. And this is actually a pretty arbitrary decision. We just had to pick a number, but basically we're saying that the heart can't really grow more than twice the mass in size, for example. But the next thing that we wanted to do was to actually calibrate this model. And so what do we actually need to do and to actually calibrate the multi-scale model? So the thing that we really needed to optimize here are we needed to be able to relate the hormone concentrations. So these are actual hormone concentrations that people have measured in different experiments. So these are in units of picograms per milliliter or nanograms per milliliter. And then also the mechanical model stretch. Solved for by the compartmental growth model into these normalized inputs, right? So these normalized inputs can take a value between 0 and 1, which are not physically like physical measurements. So we actually wanted to optimize those 2 mappings together. And so for this, we just used the linear transfer function to map. So basically like a line that relates these hormone or the network model inputs into the hormone concentrations and then the stretch. And so this is what we decided to do, but then we also needed to actually calibrate this to actual data. And so what we decided to do was to actually optimize those linear transfer functions into able to match heart growth from experimental studies of volume overload, and then also this infusion of the hormones, angiotensin, progesterone, and estrogen. And these are all actual in vivo studies that I pulled data from. So that's what I'll kind of show next. So these are kind of like the first set of results from the paper. And so for the increased level of stretch, we decided to optimize to the growth due to volume overload. And so these different symbols here represent data from all of these different studies that I collected. And then in the line, I'm showing how we can just adjust that linear transfer function for the stretch to actually match that data. So you can see that we're able to optimize that parameter in order to match this kind of curve that we observed from the experiments. And then we could do the same thing for angiotensin 2. So this is that hormonal infusion. So you can see that we can capture that nicely from all these different papers. And then again, I also did the same for progesterone, but you saw all those papers for volume overload and angiotensin. So there's one paper that I found so far that actually reports what happens with progesterone infusion. And then estrogen's a little bit better, but not by much. So there's actually 4 papers that I found of estrogen infusion in rats. And you can see that there's actually a little bit of an, I guess, atrophy, but not much. So not a very significant difference. But anyways, we can actually go through the same exercise to actually map that, optimize that linear transfer function. And then we also wanted to make sure that we're getting the loading correctly. So here I'm just showing that we can actually do that. So in volume overload, you get that increased stroke volume. So you get an increase in the cardiac output, which we're able to capture with our model. And then whereas in angiotensin 2 actually happens is it's actually kind of like a pressure overloaded state. So the blood pressure actually rises, leading to this decrease in the cardiac output. And we can actually capture that as well in our model. Whereas in progesterone and estrogen, it didn't change that much, but there's actually not too much data that's showing that. So right now we're just assuming that it doesn't have a huge impact on the cardiac output. And so again, the whole point of this calibration process was to map those network inputs from 0 to 1 to actual measurements. And so here I'm just showing what that result looked like. So for estrogen, we have on the left-hand side, the network model input. So I'm just showing kind of the relevant scale here. And then actual hormone concentrations. And so you can see in the estrogen experiment, we have this step increase in the estrogen. And then the same for progesterone and angiotensin 2. And then stretch here. So stretch is an input into the network model, but it's actually not an input into the multi-scale model. So the multi-scale model is actually calculating what the stretch should be. But the thing that we can look at here is that, as we would expect in volume overload, you get that sudden increase in stretch, which actually then kind of goes down a little bit as growth occurs. Whereas in the other cases, you really don't see a difference in the stretch, which is exactly what we would expect based on what we know about what's changing in all of these different experimental conditions. So now we've kind of calibrated the model. So the next thing we wanted to ask was, well, can our model capture the growth changes during pregnancy? So in order to test this, we implemented the hormonal changes that occur during pregnancy. So the rise in estrogen, as well as the rise in progesterone. In rats, you actually see a drop in progesterone towards the end of pregnancy, as well as a slight increase in angiotensin 2. And we wanted to see what would happen. And we also implemented changes in the cardiovascular system too. So that drop in systemic resistance and that increase in stress blood volume as well, which I'm actually not depicting here, but these are changes that we actually made in the model. And when we did this, we're actually really happy to see that we were actually able to make pretty good predictions. So here the stars are showing the experimental data from these different studies of heart growth during pregnancy. And then the line is showing our model predictions to that data. And then here I'm also showing that we can capture that increase in cardiac output that occurs during pregnancy. And so this is really comparing and making sure that our model can predict the organ level growth. But the cool thing about this type of modeling approach is you can go further than that, right? Like you can actually go in and see what's kind of happening at the cell signaling network levels. And so here I'm just showing a movie that represents the different activities of different species throughout different days of pregnancy, where pink means that it's elevated and blue means that it's going down. So you can see with progressing pregnancy that cell area increases as growth occurs. And so we can plot that with time. So here I'm just showing estrogen, progesterone, the different inputs here, and then just what happened to all of the different intercellular species. And so the big thing that stood out to us was that stretch actually did not change that much during pregnancy. So it remained pretty constant. And so the stretch in this model, again, it's an input into the network model, but it's calculated by the multi-scale model. And what it really depends on is the amount of increased loading that occurs, the rate of increased loading versus the rate of growth, right? So in volume overload, we saw that sudden increase because we loaded the heart that way. We induced that loading by introducing that fistula. Whereas in pregnancy, stress blood volume is increasing gradually as your heart is actually growing due to other mechanisms as well. And so what we really can see kind of looking at some of these curves and just the shape of the cell area, it's almost like a smoothed out version of the progesterone curve, right? Like if you kind of look through it down here. And so this was really being driven by this rise in progesterone that occurs during early pregnancy. And so that was actually allowing the heart to grow kind of at the same rate as the stretch blood volume was actually increasing in the model. And so this really suggests like an important role for this for progesterone perhaps in during pregnancy and heart growth. And I've showed this to different audiences, and some of the people that have been studying the cardiovascular system in pregnancy is actually not that surprised, because a lot of the cardiovascular changes that occurs in early pregnancy. So it's almost like the cardiovascular system is like prepping for some of this loading that's occurring. And so maybe this is a protective mechanism. I mean, I haven't fully validated this model yet, so we don't know yet, but I think these are interesting questions that we can start asking and then like designing experiments to actually like test some of these hypotheses. And so in future work, I'm really going to be focusing on validating some of these network model trends. Again, the data is really difficult to find, but I'm hoping that we can do some of these experiments with exciting collaborations. And then there's actually a particular combination that I want to test. So volume overload plus progesterone. No one has ever tested that experimentally. And so I think for this model, that's actually really important that we're getting that interaction correct. So that's an experiment that I would really love to do. And then finally, by expanding the network model more and adding more signaling pathways, because there's like at least 14 or 20 different pathways that can lead to heart growth, and really expanding that and really applying it to look at heart failure associated with pregnancy is a direction that I'm really excited to take. And so I just didn't want to stop at this, like pregnancy in general, I think is a fascinating topic. And it's not just the heart that's changing, you know, like I talked about the increase in blood volume, your lungs are changing, your bladder is changing, breasts are changing, all of your ligaments are loosening. And so I think it's like a fascinating field. And if you're a graduate student or postdoc that thinks it's interesting, I would really encourage you to explore this area. And then also just women's health in general. So like postmenopausal heart failure, for example, is a huge topic. So I really encourage you to talk to me or to other people about kind of exploring and thinking about how you can apply the skills that you've learned into this really exciting area. So with that, I'd like to thank you all for listening, and I'd like to acknowledge my lab at the University of Virginia, as well as the co-author on this paper, Jeff Saucerman, at Virginia as well, and our funding resources. And then if you have any questions, I'll be happy to take them here. Please feel free to email me or follow me on Twitter. And I'm actually working on uploading my code to SimTK, so look forward to that. And then also, I want to start a lab GitHub, so it'll be up there as well and hopefully updated so you can stay up to date with the project. So yeah, with that, I'd be happy to take any questions.
Kyoko, that was absolutely fantastic. I already prepared my students for probably an outstanding talk, given that you've practiced this through various iterations. And I think it's very easy for people to see why you ended up getting a job at a top-notch university. Congratulations.
Thanks.
Also, I want to particularly applaud how good you did in explaining all the different terms. Whenever you said something, I was like ready to type it in chat and then you would explain it yourself. It's like hypertension or high blood pressure, you know, I was like, God darn it. Anyways, thank you so much. I really appreciate it. I'm going to kick it off with a question that I had right away when you talked about progesterone and estrogen, right? In that birth control obviously uses these hormones in combination. And then actually one of my students put in the chat also a question about sort of through your cycle as well, these hormones vary. To what extent do you know or have people investigated the changes, cyclic changes as well as birth control induced changes and how they affect the heart?
Yeah, so that's an excellent question. And that's something actually I've been kind of like looking at just for fun because I'm a cyclist. And I recently learned that like depending on what cycle you're in, like your training and like your muscle function can change a lot. But the one thing about pregnancy is like during the cycle, your progesterone kind of changes like this. And then during pregnancy, it changes like this. So the levels that we're talking about here are actually like huge changes compared to the cycling. And so I've seen like some people kind of look at that. I haven't looked at that like specifically with heart growth, but like a huge topic again is postmenopausal heart failure. So before menopause occurs, women seem to have kind of a protective mechanism, especially people think with estrogen, that actually protects them from heart failure. But then after they go through menopause, they actually lose this protective mechanism. And so there seems to be something happening there. But then again, like all of these hormonal replacement therapies actually haven't been working. And so the trials actually haven't shown that they have been effective. And so I think it's still a big question mark. I think it can definitely have an effect. And I think it would be a really interesting topic and something I'm interested in actually exploring with this type of approach too. So that's a great question.
There's a question in the chat right now by Jennifer Anderson. Jennifer, I'm not sure you want to ask it yourself, otherwise I can read it.
Yeah, sure, I can ask. So along those lines, you know, we were talking about your cycle and things like that. And I wondered, okay, well, you know, the cycle is 4 weeks. It's not super long term. And I know that progesterone and estrogen can have an impact on things like breast cancer risk. So I wondered if there was any information on the effects of those hormones more long term. So, you know, over the course of a woman's life. So for example, if a woman experiences early menarche, they're at greater risk for breast cancer. Does that correspond with things like postmenopausal heart failure, or is there not really much information about that yet?
I mean, to be perfectly honest, I just haven't looked into that topic at all. And so I'm probably not the best person to ask, but from what the reading that I have done, I think there's just still a lot we don't know. And so I think that would be a super interesting topic to explore, but I just don't know, to be perfectly honest.
Lots of research to be done yet.
Yes, for sure.
Thanks.
Yep.
I had a quick question for Kyoko. So I was wondering, with the changes in heart size throughout pregnancy, is a similar kind of growth happening in some of the major vessels, such as the aorta or the vena cava? Partially also wondering because of the increased risk of stroke later in postpartum period, and if they're all shrinking back down, if that somehow correlates with that increased risk as well.
Yeah, so to be perfectly honest, I've focused mostly on the heart and I haven't looked too much into like the major vessels and arteries. But we are kind of representing that expansion by decreasing the resistance of like one of the resistors actually represents the aorta. And so that is a change that we're making, but it's kind of still like phenomenological, right? Like we can kind of get an estimate of how much is changing just by cardiac output measurements and pressure measurements. So that's how we're doing things here. But I know that it's changing a lot. So like people talk about like during pregnancy, you can actually see more of your veins because they're actually physically getting bigger. So I do think that there is a huge change. And I do think that, you bring up a really excellent point about this postpartum changes because all those changes are happening like all at once. And like your hormones, like during like pregnancy, it's kind of gradually increasing, but then like postpartum, it just like suddenly drops, like everything drops. And like all of these different changes are occurring. That's what's like contributing a lot to like postpartum depression, for example. And so I do think that that's a really interesting and important topic to look at. But yeah, that seems perfectly reasonable that those changes could be contributing to some of these like blood clot conditions, which can also happen during pregnancy as well too. So that would be a super interesting like topic to look at.
Yeah, and it's also really interesting because some birth controls, as mentioned before, have increased risk of blood clots as well.
Yeah.
Well, thank you so much.
Yeah,
and good job on your talk.
Thanks.
As a random side note, Kyoko, a friend of mine here who is in exercise physiology actually studies the effect of birth control on exercise capacity. So I'd love to introduce you. I think there would be a lot of overlap. And she actually recently published a paper that shows that birth control actually enhances your oxygen carrying capacity. So
oh, really? Okay.
Just random side note. Also, there is work by Sarah Wells who looks into pregnancy-induced heart valve growth. Yep. Which of course is pretty dear to my area of research, just for people that are interested in that as well.
Yeah, I love her paper. She has this really cool paper looking at the mitral valve in cows, and like they grow tendinae, which is actually kind of crazy, but like that image will forever be in my mind. Like I love that image from her paper. So yeah, for sure.
It actually freaked me out so much that I investigated it more and I talked to her on the phone, and it seems like the cordia is splitting. So it's one cord that splits in half and then kind of like translates over. So it's not like these ropes grow, but nonetheless, it's incredible. Yeah.
Yeah, for sure.
Any other questions by my students? Oh, Jacob actually has a question here. Jacob, do you want to ask the question or do you want me to read it from the chat?
Ask. So do the hormone changes initiate only growth? So like in cardiac regenerative medicine, we're trying to like re-jump like cellular replication of the myocytes. So how can like the computational modeling framework do stresses and hormonal changes if we're able to initiate that cellular replication? So is it finite enough to understand like this is due to an increase in cells rather than an increase in I guess like protein concentrations?
Yeah, so that's a really important, I think, distinction. So right now in my model, because we're looking at like the in vivo state, we're just assuming that everything is due to the cell changes. So not like proliferation that is changing within in vivo, which I think is a pretty good assumption to make. But like, I think we can get even more mechanistic than we are now, right? Like we can actually like look at which proteins are actually changing within the cell that's leading to this increase in cell area. But I think in terms of like regenerative medicine and myocytes, I do think that we can, I guess it depends on like what question you want the model to answer. I can definitely see like if you're interested in some of the signaling that's actually leading to these changes, like you can actually take in some of these interactions and actually just look at the systems model, for example. But I'll say that like one of my plans in my lab is to actually take the same framework and apply it to the uterus. Because I think the uterus is a fascinating organ that like no one is really like looking at too much. And in the uterus, we know that in early pregnancy, we do have this proliferation that occurs in the uterine myocytes and then hypertrophy that occurs. And to be honest, like I haven't thought about too much about like whether or not we can use the same like growth changes to represent both in terms of the mechanics. But I do think that like some of the signaling must be different too, right? In that proliferation versus hypertrophy. So yeah, I haven't really decided like what exactly it represents, but I'm hoping to explore a lot of that, more of that like in the future.
Fantastic. Any other questions?
I have a quick question, if I may.
Yeah.
Yeah, great job, Kyoko. Super interesting stuff. And like Emmanuel said at the beginning, it's really neat to see you sort of synthesizing your PhD and postdoc work in a way that's like very impactful. So
thank you.
I did have actually kind of a follow-up question to what Jacob just asked. So in terms of the cellular responses, I was wondering if you've looked at, and maybe you came across this in the literature when you were building your network model, but which cells in the heart actually express the estrogen receptor and the progesterone receptors? So is it an effect directly on cardiomyocytes, or is it cardiac fibroblasts somehow? Or if you could comment on that.
Yeah, so that's also an excellent question. That is still a huge question mark for me. So estrogen is particularly interesting because a lot of people have been studying that. And there's actually like a debate going on in the literature from what I've seen, whether the estrogen receptors are actually on the cell surface or in the nucleus, like on the nuclear envelope. And then in terms of, I think there's estrogen receptor alpha and beta. Some people say only alpha is expressed in cardiomyocytes, and other people say that both are expressed in cardiomyocytes. I forget if it was alpha or beta, but there's still this like huge debate going on whether like which receptors are actually expressed in cardiomyocytes. And so the answer is that like that's why I didn't put the receptors on there. So I do have an undergrad that's like trying to look at some of that signaling just by looking at estrogen, just because there's just more data there. But yeah, that would be something that I'm hoping that the people who are investigating that will kind of figure that out soon. But it's kind of fascinating because like that seems like such a simple thing that we actually still don't know the answer to. So yeah, the short answer is we don't know.
If there's not another question, I have another one that's completely different. So I don't really study the heart all that much. I mean, a lot of my background is in vascular disease, and there's sort of this, running theory that estrogen is a protective hormone, quote unquote, protective hormone against a lot of like, particularly mouse models of vascular disease. And that the counterpoint to that is that testosterone is probably more detrimental to promoting disease. So obviously outside of the context of pregnancy, have you thought about the role of testosterone in any of this as another, sex hormone that might be promoting some of these changes in the heart?
Yeah, I actually haven't looked at testosterone. I've actually come across papers that look at estrogen, both estrogen and testosterone. But something I just haven't focused on just because, I don't know, I guess because I'm one person and just like the manpower of like just sorting through that data. But yeah, I do think that, if we wanted to apply this for like postmenopausal heart failure, or even just, this doesn't have to be for the heart. It can also be for, the arteries and other muscles as well. I think that would be interesting and probably important to look at some of those signaling pathways too.
Awesome. If there's no more questions, I would definitely release my students into, their free world, but also everybody else on this call. But I don't want to take the opportunity if there is one or two more questions. I have time, and hopefully Kyoko also has a couple more minutes. One last opportunity.
Yeah,
here we go.
Someone else.
I have a question. When you have a network model that characterizes some physiological phenomena, how do you make sure that the way that you calibrate these network models is actually representative of the exact, like whatever is happening at the cellular levels, just by looking at observations on like a much higher scale?
Yeah, that's a great question. And so the way I kind of calibrate it, I didn't really actually calibrate the network model. So what I did was just look at, if I increase one input, what happens to the output? How much does the output go up or down? So the cell area go up or down? And then I looked at combinations. So if I increase angiotensin and estrogen, what happens to the cell area? Is it higher than angiotensin alone, or is it lower than angiotensin alone? So that's kind of how I calibrated it, but that calibration is just looking at the connections and then looking at the logic gates. So you can have AND gates or gates in that model. And so that's kind of how I calibrated it. But one of the advantages to this like normalized approach is that the effect of each of the inputs just depends on how many connections you have. And so you don't necessarily have to calibrate it. And I would say that right now we just don't have the data to actually say like, this particular reaction takes up more of an effect than another neighboring reaction. And so simply we just don't have that data. But I do think that is something like once we can look at get more data, then we might be able to actually calibrate that. And I know that, in some of the other work that other people have done, they're actually able to do that. So some of these like single-seq RNA experiments, for example, they're actually able to kind of look at some of these levels. And so I do think that the field is actually heading in that direction. But for this one, I just basically said, okay, it goes up or it goes down. And that was the amount of calibration that I did for the network model.
Oh, okay, thank you.
Yeah.
I think Johannes, you also had a question, right?
Yeah. It's more of a sort of general picture question. You motivated your talk with saying that heart failure is one of the predominant causes of death to women in pregnancy. Can you just from how would you continue to use your model to arrive at some clinical relevance in order to understand this problem better?
Yeah, so the condition that I'm really interested in is peripartum cardiomyopathy. So this is heart failure that occurs towards the end of the pregnancy or after the mom gives birth. And so a lot of people have been focusing on certain hormones that they think can actually affect or like predispose women to this condition. But the problem right now is that we don't even know like which woman is going to have experience this. And so it's actually a really sad condition because, here you have a woman in her 20s and her 30s. She's a brand new mom. She gives birth to this beautiful baby. And then her heart just doesn't go back to normal and she has to go on a left ventricular assist device and then wait for a heart transplant. And so what I'm hoping to do with this modeling approach is that I really do think that it's this combination of all of these mechanical changes and these hormonal changes that are occurring so quickly towards that stage of the pregnancy. So I showed that cardiac output curve, right? So you get this increased loading and all of a sudden you get this unloading. And so as a mechanical engineer, I think that that's really important to look at. And so I really want to test some of these hypotheses that these interactions are actually important. And so that's kind of like the direction that I want to go. And I'll say that there's been a lot of development in animal models for peripartum cardiomyopathy. So I think it'd be interesting to look at like, okay, if we think this hormone has this effect, can we actually manipulate some of the mechanics in the heart? And then make some predictions with our model and see, you know, does that affect heart failure in a way that's good or bad, for example. So that's kind of like the big picture way of looking at it. And then like once we expand the network model, we can actually start looking at, you know, what happens with all these different like genetically modified mice. So we know that some mice have different types of hypertrophy, like just during development. I think it'd also be interesting to kind of look at with pregnancy and what happens to these mice with pregnancy, and kind of, you know, alter the hormones or alter the mechanics in some of these animal models to really start understanding these mechanisms that could perhaps be leading to some of these heart failure conditions. So that's kind of like the big picture image that I have for this type of modeling approach.
Awesome, thank you, Kyoko.
Yeah.
Shall we call it quits here? I know I said there was one last opportunity, maybe one last opportunity.
Yeah, I'm also happy to just like stay on for another 9 minutes.
Very
or until
cool.
noon. So
I guess I have a question. Really
Oh, there
quick.
we go. There we go.
First of all, thank you for a great talk. I think it was very interesting. My question pertains to, I guess, the disparity between maternal outcomes here in America. I think for the most part, it's really clear that disparities in access to healthcare and quality of healthcare results in Black mothers having much poorer outcomes. I guess from your point of view, I wanted to hear your opinion on this, but is there any sort of effort that is assessing maybe biological differences, like ethnic specific differences in responses to pregnancy or responses to like hormones or anything like that?
So I haven't looked at that like papers specifically looking at that yet, but I do think that it is important, and I do think that that is something that we can start looking at with this type of approach. And it's not just pregnancy, right? Like if you look at even Black women are just at higher risk for heart failure just in general too. And so I really do think that it would be really cool to take this field that way. But then again, like we need to know what kind of like biological data we need to be looking at. And so I'm hoping that, you know, with this push that we will be able to start answering some of these questions. But like one thing that I think would be really cool is, you know, like my PhD advisor was one of the first people to dedicate her research to women's health, and I'm dedicating my lab to women's health. But I'm imagining that, you know, maybe like one generation from now, so maybe one of my grad students, if they wanted to kind of take this type of approach and really focus on that and like really looking at some of these health disparities, I think that would be something that's like really exciting. And I'll say that there's actually a lot of funding in that right now. So NIH has this initiative called the Improve Initiative. And so hopefully, you know, if we invest money in looking at some of these and really encouraging people to really explore it, I hope that we can look at it a little bit more biologically. But but right now I would love to do that. I just don't have the training. But if you do, and you're interested in using my model, I would love to talk about it. But yeah.
Yeah, thank you.
Mm-hmm.
I think, Kyoko, we're going to release you here. I think you did a fantastic job. I think this was by far the most questions anybody has received and answered. So we really appreciate all your insight and you patiently answering all of our questions. Yeah, thank you, everybody. Appreciate it much. Wonderful talk, wonderful attendance. And yeah, stay tuned for our next Journal Club.